I spent some time this afternoon trying to actually calculate something on a whiteboard for you and demystify the process. And I realized that at times it may have gone a little bit too fast. But my point really was that literally we were able to do a non-trivial calculation in 1 hour, less, half an hour on the blackboard, right? And if you had any trouble following it in that half an hour, if you spend another day, you will have it. So it is completely within reach, right? And do not be intimidated by this. But now I actually want to tell you what all of that effort, all the algebra, arithmetic, was aimed at. And well, there's actually quite a distance we can go with it, but we don't have time. So I really scaled down my ambitions, but I still want to get you to some logical conclusion. And I thought what we can do is just go back to a couple of results that I've already advertised to you yesterday and work them out. And one result is calculating this rate of adaptation in a large asexual population where many mutations come in more or less simultaneously and compete with each other. And as I explained yesterday, under these conditions, the rate of adaptation is no longer linearly increasing with mutational influx, but instead grows very much more slowly. And then conventional wisdom goes that in the presence of sex where recombination can put beneficial mutations together, one can restore linear dependence. But so we're going to actually calculate and confront this conventional wisdom to more serious analysis, and we're going to learn that it isn't quite true. So, but let's first redo this calculation for the rate of asexual adaptation. Basically, this was worked out by Michael Desai and Daniel Fisher merely, 6 years ago. It's one of those surprises. It is addressing such a simple and classic problem that you would have thought was all sorted out 50 years ago. But this is actually turns out to be quite recent. So again, the reality is complex, and the idea to make progress is always to simplify and keep as little as necessary to get the gist of the phenomena. And once you understand what's going on, you can add bells and whistles, make things more and more complicated, and you're going to perhaps find out that a lot of complexity can be incorporated without really changing the result very significantly. And then there'll be certain things that will actually turn out to be very important, and you will have to move beyond it. So let's start with the simplest case, where we'll consider only beneficial mutations and make all mutations have the same size, right? And then perhaps there are also a lot more neutral mutations, but they don't bother us. So a single mutation advances the fitness by a little step. So mutations move individuals from some fitness bins to the next bin. And then, of course, selection, we know, is moving this whole distribution forward. And we're particularly interested in what's happening to this maximum fitness to the top bin as the distribution is sliding forward. Well, the very general statement that we can make is the following, that mutations are coming in with this rate into the population, right? Mutation rate times population size. Each one of them is advancing the mean, is bringing with it fitness benefit S. And then the question is, what is the probability that this allele will fixate? What is the probability that this allele will find its way into the whole population? And that's the non-trivial factor, right? If we didn't have a broad distribution here, if we had just sort of a single clone, right? Imagine that we didn't have this whole thing, and the whole population was just sitting in one bin over here, then the probability of fixation, we calculated it's still on the board here. There it is. Will be proportional to its fitness benefit, delta x. Delta x for us just became S. So this whole factor would just be equal to S. And we would recover exactly the situation of low mutation rate, small population, where there is no diversity, right? But now we have a little bit more work to do, right? So this is mutational influx. So the key insight of Michael and Daniel was basically to realize and actually to demonstrate, so I'm going to skip the explicit calculation, that basically because of the competition between the clones, only the fittest individuals here, only mutations occurring on the best, very best genetic backgrounds, those are the only ones that are going to survive, right? So basically, instead of mutations accumulating in the whole population, only the influx of mutations into that top bin matters. So we have to reduce that factor of n by the fraction that this fittest subpopulation represents, right? So n nose, I guess this was a nose, over n. And in addition, there is another factor corresponding to this rather small survival probability and high probability of extinction, right? This nose still represents a relatively small advantage compared to the mean, and not all the mutations that occur, even on the best backgrounds, not all of them are going to fixate, right? Only these ones. And then off we go. We need a little estimate of, so how big is this leading population? So now we go back to this traveling wave business, and we know that this leading population, not so long ago, was just this single mutant. It started with one, and but it's an advantageous mutation. It's a fit genome, so if it doesn't get extinguished, that's the factor, it's going to grow exponentially with this growth advantage. So somewhere along the way, I lost deltas here. So my x's here really should be delta x's. So everything is happening relative to the mean. But of course, it's growing exponentially with time. The question is, what is this time? What's the relevant time? And the relevant characteristic time, of course, is the time that it takes for the leading bin to become a subleading bin. The time it takes for the new and better genotype to arise in the population. And that time corresponds to, what the hell is that? Sorry, something's missing. Oh, no, nothing's missing, sorry. Yeah, that's fine. It's just that I can't see it. Can you see it? So mu times this exponential basically has to be over the 1, right? Because this is so the probability per, to generate a new, to seed a new clone. And so if this is 1, I'm actually being a little bit sloppy with logarithms. So strictly speaking, it should be, again, I want to simplify a little bit. So we can essentially forget about this factor, right? This is 1, and this is all we have, particularly simple. And all we need now is to remember that the fitness of a leading edge relative to the mean, remember we worked it out today, depends logarithmically on the population size. And we have pretty much everything we need. Right? We know what the leading edge, the fitness at the leading edge is relative to the variance. And but you will say, well, but we don't know what the variance is. But then Fisher's theorem actually tells us what it is, right? It is equal to the velocity. So we have yet another equation to combine with the one that we have here. And now we can essentially eliminate one of our unknowns, the sigma, and recalculate the whole thing, recalculate the velocity, and we're basically going to find that it only depends logarithmically on the population size. And in my eagerness to simplify the argument, I have lost some logarithmic prefactors here, which actually reintroduce the dependence on the mutation rate, but in fact very, very weakly, right? Mutation rate only enters through these logarithms. And the bottom line, the rate of evolution in this genetically diverse population is very much reduced compared to what it would have been in the clonal population, basically because most of the backgrounds, most of the genetic backgrounds, except the best or very nearly the best, are bound for extinction. They are the typical genotypes in the population. But they're in the dustbin of history. They're living dead. And right, by sampling this population, it will be pretty difficult to actually uncover the genotype ancestral to the future generations. It is an outlier. It is atypical. But everything, all the non-trivial so the non-trivial part of the story then was hidden in this survival probability, the probability that the genotype does not become extinct. Okay. Yeah, well, it's just a logarithm. Sorry, I use different prints, but it's the same logarithm.
Log squared.
Yeah, log squared. Sorry. Yeah, oh, I'm sorry. Yeah, ah, boy. Yeah, a logarithm of this thing squared. Sorry, I wasn't sure. Yeah, I don't even know what sort of the authorized notation is anymore. Okay. So the whole story was about interactions, but we're going to skip the small talk and now confront the same problem, but in the presence of recombination. And let's think through it again. And now I will use a little bit different pictures, and I think that may actually be advantageous, for you to think basically the same process, but a little bit from a different angle. So, okay. So here we have this fitness distribution, and a new mutation occurs somewhere on some background. And this fresh mutant is better than average, so it starts growing, starts expanding in the population. So the size of this clonal subpopulation is growing. So let's say time is going this way. But remember, we're thinking of a case where mutation rate is high. So other genotypes are also begetting good mutations. And the whole distribution is improving, right? So if all we have is, if our, the progeny of our mutant is just clonal, then eventually they're going to find themselves not ahead of the mean, but behind the mean. They got overtaken by other genotypes, right? So this population will start collapsing, right? So this is precisely the living dead situation. They are on their way out. Unless some member of this clone can recombine with some other genome. And if it marries well, then it can move onto advantageous background, right? And start a new clone, which is better than average, and it will start expanding. So our polymorphism, therefore, persists, although on the new background, right? And in order to survive, of course, in the long run, this polymorphism has to hop again and again and again to stay at the leading edge of the front. So think of surfing, and you have to stay on the front of the wave, and if you fall behind, you're gone. So right. So another way of thinking about the situation is, of course, well, I'm just replotting. So here is my mutation, and the clone generated by that mutant. It is expanding, and now there are these recombination events which are going to start new clones on different backgrounds. And some of them may be good backgrounds, but most of them are probably bad backgrounds. And we can think of this as, how am I doing on time? Okay, so we got the clone. So this is our fitness. This is time. And I'm going to represent the existence of this clone just as a stick, sort of a line propagating in time until this clone becomes extinct. So if that's what happens, the clone is extinct. But along the way, it has recombined on different backgrounds, started new clones. And every one of those did the same, and so on, and so forth. And right, this whole collection of clones, this whole collection of sticks now represent this sliding traveling wave moving forward. And our calculation, therefore, has to reconcile this stochastic picture of generating new clones and new backgrounds and kind of deterministic picture of Fisher traveling wave. Genetic background. So we have a genome, right, and mutation occurs on it, right? But so we're referring to the genome as the genetic background, right? Because we're talking about diverse population, so there are many different genomes represented in it, even though not uniquely, because right, we're in a regime where there's still clonality. There's some probability of finding two individuals with identical genome, right? But still, there are many, many clones competing, right? And I'm referring to, and they would have, right, and different clones, we say, have different fitness, and right, mutations can occur in any of these clones, and right, then, of course, generating yet a new genotype, starting a new clone. So that's exactly the process that we're talking about, right? So if we cut, look at the so the population at any given time, there is this bunch of clones that are competing, and these are different backgrounds. So when a new mutation comes in, it can occur on, right? So suppose a new mutation comes in at this time, it's going to hit any of these things, right? Okay. So you can simulate this very nicely, and we're basically interested in so the probability that so a new mutation, maybe you can't quite see the sticks over there, will spread over different genetic backgrounds until it is essentially found in all of the clones. Right? At this stage, every member of the population has the polymorphism it has fixated. Right? So the game for a given allele in recombinant in the sexual population is to diversify and spread onto all of the backgrounds. And the question is it going to succeed, right, and be present everywhere? Right, so basically, if I now cut across at a given time and look at so the density distribution across, I'll get the same Gaussian lump. It is everywhere. Or in most cases, it will become extinct. Right? So we're going to do exactly the same calculation as before. Right? This is sort of a nicely general and universal description of the process. Is just that calculating this survival probability has to be done including the recombination process. And the thing about the recombination process, it allows a given allele to spread over many different backgrounds. Right? There is nothing better than this sort of diversification. So if you want to think so intuitively of the benefits of sex, is, right? So think of this selfish allele, and how is it going to survive? We're now talking about very simple sort of steady state wave, constant selection, constant environment. But in general, there is no such assurance. So the best way to survive is to spread over all possible genetic backgrounds. That's the name of the game. Let me walk you through this little calculation now. So we got a new clone started here, and with some largest probability. So the way we think of fitness advantage here, it is small. We're talking about effects of few percent. So with probability rather close to 1, it's just going to become extinct right away. But with the probability 1 minus that, it will not go extinct, at least right away. And it's then going to generate these lots of babies, recombinant babies, on different backgrounds. And the number of offspring here is basically going to be Poisson distributed with some rate, which is proportional to the size of this clonal bubble. But now the key of the calculation is, so basically until I introduced this, this was a very fancy way of writing 1, because this whole thing is 1. But now what we're calculating is the probability to become extinct, right? And the way we're calculating it, either this allele became extinct right away with this probability, or if it didn't become extinct right away, then all of its offspring babies have to become extinct. Right? And now we actually have an equation that we can work with. So of course, right? So this became a closed equation. So we related the extinction probability for an allele here to an extinction probability of all of these baby alleles, which are of course governed by exactly the same function. So the only thing is we have to average over all the possible backgrounds that they can sit on, and that's what our model of recombination and what it does to fitness is going to provide us with. And I guess last but not least, we can calculate the size of this bubble basically by saying that the size of the clone grows exponentially, except eventually when the mean starts overtaking it, it collapses. So a little homework exercise. So that's it. This is the calculation. We got ourselves an equation that we can solve, right? Because okay, sorry, I'm still trying to go a little faster. All right, yeah. Right, we have our very general expression for the speed of adaptation, and we just outlined, at least wrote down the equation that governs the survival probability. Survival probability depends on the variance, of course. It depends on the rate of recombination. And then we have another equation relating the variance to the velocity. Thank you, Mr. Fisher. And now we have an equation that actually allows us to calculate the variance, just looking at this equal to that. And remember, that was exactly our job in with Fisher waves, right? So we had this simple Fisher's traveling wave, but in fact, it could propagate with it could have any variance whatsoever. And now we have an equation that allows us to calculate the variance, and therefore we know velocity. And that is a result. So what we see now, the rate of adaptation increases actually quadratically with the rate of recombination, with the frequency of outcrossing. But at least in the limit of so large mutational fluxes, we do not recover the linear dependence as sort of promised by this common wisdom. Instead, we still have this only logarithmic returns. And the way this, of course, works out is there is a linear regime that harks back to low mutation rate, low mutation flux regime. And now that linear regime expands further and further out to higher fluxes before it crosses over to a logarithm, right? So for a given mutational flux, you only gain logarithmically with increasing population size. No, I'm sorry, no, for a given mutational influx, you gain as r squared by increasing mutational rate. But for a fixed rate of recombination, you only gain logarithmically by increasing mutational size, mutational influx. So, and that's the kind of thing that I promised we were going to do. We were going to calculate, we're going to study sort of functional dependencies of the rate of adaptation, for example, on the population size. And going back to yesterday's background introduction, the reason you might be interested in functional dependencies like this is when you're, for example, trying to figure out how the rate of acquisition of drug resistance in HIV, in AIDS, depend on the viral load. And if you're applying the drug, you may want to know how much you want to invest in bringing the population size down. So that's sort of general logic. Yeah.
So there is a need, right? So it changes
Yeah.
behavior from linear to that.
Yeah.
So but your equation doesn't tell us right now where the need is.
Yeah, it doesn't, but you can figure it out very easily. Well, you can sort of calculate a little more, but you can figure it out very easily just by comparing what this thing says with what that thing says. And that will give you the crossover
So in HIV,
pretty much correctly.
So in HIV, where are you? Are you in the R limited? Is R high enough so that your
Well, it depends on n, right? But you are in a limiting regime. And I think you're probably tired enough that we should stop more or less here. And I didn't put anything on the slide because I didn't have time. But I just put it up there so that I would know to talk about it. Anyway, so I could really tell you more about various things. But this is a good a stopping point as going to present itself in the near future. So I'm going to stop right here. So I just presented you with these couple calculations which dealt with sort of the rate of adaptation in a diverse population. And there was a certain beauty to it, that it was actually a single, rather simple idea that got us through both asexual and sexual case. And you may say, well, but we only looked at the beneficial mutations. And we are talking about some constant environment and steady state waves, and this is some theoretical idealization, and the reality is not like that. Well, so in fact, it's sort of amusing. So every time you I guess Rob complained about reviewers, referees, so I can complain too a little bit. There is an obviously very strange thing that we're doing here. Well, in fact, going back to Fisher, we're saying that mean fitness is increasing linearly with time. So if you think of fitness as brood size, it obviously doesn't increase, with time. So something a little bit silly is going on. But on the other hand, now think about deleterious mutations, right? And with very crude numbers before, we said that deleterious mutations are at least 10 times more frequent than the beneficial ones. So as we're sort of happily calculating here the traveling wave moving forward, we also have to think about this influx of deleterious mutations that is constantly pulling the population back. So, and the beauty is, with this machinery, we don't have any problem incorporating it whatsoever. So now just think of running up an escalator moving down. So if it weren't for the beneficial mutations, deleterious mutation would have accumulated and wiped out the whole thing, right? And so this is your Red Queen scenario, that the population just has to run forward, adapt as fast as it can to just hold back the influx of deleterious mutations. And with SID, we actually worked out this quite explicitly. So now suddenly what this whole story tells us is not just about this directional selection and continuous adaptation. It is suddenly telling us something about a rather stationary, sort of evolutionarily stationary population. And so if we didn't know anything about the genetic sample that we took from the population, the question is, what assumptions should we make about that? So one possibility is to say that, well, all this variance and all the variation that we see in the population is neutral. It doesn't matter, right? And that has been and still is the null hypothesis that all the interpretation of genomic data is based on. And we're saying, well, perhaps instead we should be using as a null model this model with fitness diversity. And we now have a pretty decent understanding of data, and which is done basically by looking at the structure of genealogical trees. So, you know what? I can do it in 5 minutes. I can do it very crudely. No, wrong way. It takes its sweet time, huh? So let's think about the genealogy of the population. And let's start with an asexual population. So remember some of the pictures that I drew for this Wright-Fisher model. And at that time, we were thinking about what's happening in the population going forward. Now let's think about what's happening in the population going backward. So suppose you have 2 individuals, and you ask, who are their ancestors in the previous generation? Well, with high probability, their ancestors are different. But there is a chance that maybe these 2 individuals are sisters. And then you do it again. And I'm going to skip to fast forward, or rather fast backward, because we're going backward in time. And what you're going to see is a genealogical tree, which takes us back to the common ancestor. So in a neutral theory, which, as I say, is currently the basis for all interpretation of the data, we know everything about the statistics of this genealogical tree, which is called coalescent. There's a name. Kingman's coalescent. And for example, we know that on average, the time for any 2 samples to come to a common ancestor, that time on average is equal to the population size. And on average, the height of the tree, t to the most recent common ancestor, on average, is equal to 2n. So this last coalescent event takes half of the time to the common ancestor. And more recent, so the earliest coalescent events are relatively much faster. And again, the whole thing can be derived in 15 minutes, and I was going to do it, but I'm not going to take 15 minutes, so just take my word for it. But the reason this is interesting and important is if you want to estimate the mutation rate, right, in this genealogical tree, you have to think of mutations as just random events which occur with some Poisson rate, mutation rate, along this lineage. So one way of calculating the mutation rate is just look at pairwise differences. For example, look at this pair and ask how many polymorphisms distinguish them, right? 1, 2, 3, 4. Do it for all the pairs. Take the average, and right, this average genetic distance is mutation rate times average time. We can measure this. We know this if we know the population size. So we just got mutation rate. Or we can ask how many different polymorphisms do we have in our sample? Right? For that, we need to calculate the total length of the tree and count up the total number of the events, right? And then so the number of what's called segregating sites, segregating polymorphisms, is basically going to be mu times well, twice T2 times the logarithm of so the size of the sample, the number of samples that we're looking at. So that gives us another way of estimating mutation rate. Given, right, this whole statistics, however, is based on the assumption of neutrality, right? And there is a simple test, therefore. One can just take these two estimates, look at pairwise differences, and look at the total number of segregating sites. We have a theory, right, Kingsman coalescent theory, that relates the two, right, in terms of this logarithm of the number of samples, right? And we can compare, and if they agree within, of course, one has to think of statistical deviations, which again one can calculate all the variances here, right? So if you have 3 or 5 sigma deviation between two estimates, then you'll say this is not a neutral process. There is evidence of selection. So that's kind of a state of the art until recently. But what we've learned to, we, meaning collectively a bunch of people working in the field, what has been learned recently is, and I guess the relevant names here, Bernard Derrida, Richard Nair, Oskar Halachuk, Daniel Fisher, Michael Desai, what am I forgetting? Well, and friends, and company. We basically now understand what the coalescent process should be, quite generically, for the population with fitness diversity, right? So instead of referring to this neutral null, now we can interpret the data in terms of this alternative null, right? And curiously, the genealogies in population under selection are remarkably universal. And they look very different from these trees. And the last tidbit that I was going to maybe tell you about is that one can push this further still. Once you realize that the shape of this genealogical tree contains signature of selection, right? Not only you can detect from statistical properties of the tree that there is selection, but you can actually try to identify branches of the tree that have higher fitness than the rest, right? So we can try to use this to actually predict the future. And that's sort of very much where we are at. And my extra hour is up. So I'll just, there you go. No. Very slowly it unfolds and comes into focus. So Richard Nair and Sid, some of the partners in crime, at least for the parts of the story that we are responsible for, which I guess I didn't talk that much about. Well, I mentioned what we did with Sid on so dynamic balance of positive and negative mutations, also with Richard. And with Richard and Daniel Fisher, we did this calculation of the rate of adaptation in recombining populations. And Adel is one of the current postdocs at KTP, and we've been fooling around with these attempts to predict the future from genealogies, and it is somewhat possible in silico. So we'll see where that takes us in reality. So thank you. Mm-hmm.
So as you also said earlier in your talk, right, that because it's a Gaussian distribution, whenever you will go and sequence these things, you'll end up actually picking the guys from the living dead.
Yeah.
Right? Because the guys who are actually going to be in the future are actually literally very buried in the tail, which you will never quite sample.
Right. So what you're trying to, So what you're trying to identify, right? You will never catch the ancestor of the future, right? The future progenitor, right? But what you can try to identify is the common ancestor, the closest living relative of of that progenitor. Right? So the game is now if you have a sample, and from much larger population, so there is a lot of stuff that didn't get sampled, and right. So and suppose this guy is actually the future, and this is your sample. But everybody comes to the common ancestor. So this guy also comes to some common ancestor of your sample. You have not sampled this branch, right? But you can see into the future, into the past from the sample, right? And then the bottom line is, can you, so basically, identify this clade, which are the closest relatives of this guy. So they happen to be, second cousins here, right? And, right? And they will be much more similar to this genotype than those guys. So if you can identify that much, you've learned something. Not everything, but something. To figure out, and we have a little algorithm that does it for you. Well, yeah. So basically what's happening is, in fact, the trees under selection have this sort of peculiar property right? These things have, I said that so the deepest branch is the longest. And genealogies under selection, actually, it's the bottom branch. So they look basically like combs, right? So it's the bottom, most recent legs that are the longest. And that basically means that there are a lot of mutations that arise just at the bottom. And then these mutations are private. These are singletons. So you will have a hell of a time detecting them, right? So the information is contained deeper in the tree, right? And so the most informative layers are somewhere, halfway through the tree, right? And now, of course, if you're in a situation where it's just a single big mutation that makes a difference, there's nothing we can say, right? It's one of these things, and we have no information about it, right? But if you are in this statistical genetics regime where it takes many mutations, accumulation of many mutations with relatively small effects, then there's something you can say. But I haven't told you how. Okay. More questions? Yes? No, 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 no. The deleterious mutation can also serve to fixation. Deleterious mutation can also. Hmm? Yeah.
But supposing you do not have recombination, there is a possibility that another mutation can arise, and the combination of the two mutations can become beneficial.
Yeah, so that is fairly unlikely. So yes, and that will happen especially if you get some reasonably high probability a very large effect mutation, right? So if simple Fisher model where mutations were all the same size, it basically doesn't happen, right? But if you allow longer jumps, and especially if you say that the probability of having a large jump falls off more slowly than exponentially, then so the tail of this non-extinction probability starts extending deeper into the bulk of a distribution, just as it did in case of recombination, right? They're still very bad, mutations that fell on bad genotypes, bad backgrounds, still had very small chance of surviving, right? That's sort of why we didn't get didn't recover the linear dependence on the flux. It is still localized at the leading edge, but so the width of that leading edge was increasing with the rate of recombination, right? So similarly, if you introduce these sort of large effect mutations, then they can start carrying sort of passenger deleterious things along.
Second question, in certain cases, the mutation rate itself can be subject to selection.
Absolutely. Well, I mean, it's not sort of a different case. You just you have to ask yourself, where did these evolutionary parameters come from? So, well, one can take this point of view that when we talked about it yesterday, that the less mutation, the better, and mutation rate is basically set, is as small as it can be. Well, but there are mutators, right? And they seem to be epigenetic mechanisms to actually turn on the mutation, right? And then you can ask, well, there are mutated alleles surviving in bacterial populations, and why they're surviving? What's the probability of, is there an optimal mutation rate in the population? And you might expect that in a rapidly changing environment, the organism will want to crank up the mutation rate. So, and again, yesterday we talked about, so how does one begin to think about selection acting on these, like secondary traits, on these traits that do not directly contribute to the fitness, but if they do, then perhaps negatively, right, over the short time. And again, the way this picture presents a perfectly good handle. The most important thing is non-extinction probability, right? We were just calculating this non-extinction probability in the simple cases, but you can generalize this and now say, what is the non-extinction probability in a time-dependent environment? There's no problem in calculating it. The only problem is in figuring out what assumptions you want to make about the time-dependent environment. But if we can agree on the assumptions, we can calculate it.
Isn't this estimation of mutation rate the estimate of only beneficial mutations, or and the actual rate of mutations?
Oh, no, no, so, yeah, so that's actually all neutral. So I started, what I was writing on the blackboard here was all a neutral picture, right? And yeah, so
Because
then when I start talking about these genealogical trees with selection, again, you expect most of the mutations that you see, you still expect to be neutral, right? It's just that enough of them are non-neutral to generate the fitness diversity. But it is sort of a safe assumption that, at least, that the ratio is, 10 to 1 or maybe 100 to 1.
so the actual mutation rate would be something different than what is estimated, isn't it so? Which contributes both beneficial and deleterious mutations? Because the sample size itself for a deleterious mutation wouldn't be available.
Well, no, you see, right, in this picture, we don't know which mutations are beneficial or deleterious. You are seeing all of the polymorphisms, right? They all show up in the genome. And all you're doing is you count how many different polymorphisms you see in the sample. So there are two big issues here. Two big issues. One is if the mutation rate is so high that you start getting the same polymorphism multiple times, right? So then you can start undercounting the mutation rate. And another big can of worms here is, so I said that we know the population size. Well, actually, it's very, very difficult to know the population size. Certainly in most field situations, and that's what evolutionary population geneticists and ecologists argue over. What is the population size? And there is a notion of an effective population size. And to complicate things, these sort of passenger interaction effects of the fact that okay, so the presence of selection can to some extent be subsumed, or one might try to account for the fact that these trees under selection actually come to the common ancestor much more rapidly than the neutral trees, right? These trees are shorter. And then one might say that the relevant population size is not the census size, not the number of individuals, but some effective population size. And that's sort of a terrible fudge factor. And so figuring out a better way of dealing with the conundrum of effective population size is sort of the driver for actually understanding these different genealogies and the effect of selection and so fitness diversity in population. So we want to understand that.